Hello, everyone. Today, I'm going to talk about plastics, their toxicity, and how these toxicities are modified in the presence of other contaminants. Plastics are everywhere. They are in every single element of our lives. From the time we get up in the morning until we go to bed, we're exposed to thousands and thousands of plastic particles daily basis. Of course, these are fantastic materials. At the same time, they are toxic. I'm sure we all have seen these kind of heartbreaking pictures that alarm plastic threat towards the wildlife. Possibly, these could be the ones that started the whole movement of public awareness on plastic toxicity. Plastic comes in different shapes and different sizes. Macroplastics and their toxicities are extensively studied. Once they get into the environment, they break down into smaller pieces, which are known as microplastics. Microplastic toxicity has also been studied quite well. If this degradation continues, they further break down into finest particles. Scientists now call them as nanoplastics. The study of nanoplastic is relatively new, and our knowledge is limited. Therefore, our research team started to investigate the nanoplastic toxicity on an organism. They are biologically active. Once they get into the body, they can have negative impact on physiological processes of an organism. Earlier in our lab, we exposed zebrafish embryos to polystyrene nanoplastics and studied for the uptake and distribution. We had 3 concentrations, 0.1, 1, and 10 ppm of nanoplastics. Nanoplastics were taken up by zebrafish embryos and mostly accumulated in the lipid-rich areas such as yolk sac and the brain. When nanoplastics are in the natural environment, they bind to other lipophilic compounds such as rotenone, which is a pesticide and a potent mitochondrial toxicant. This sorption can either increase or decrease the toxicity by expediting or limiting the uptake. Mixture toxicity studies are important in assessing ecological risk. In the current work, we investigated the single and combined effects of nanoplastics and rotenone on bioenergetics of an organism. Zebrafish embryos were exposed to 3 conditions: nanoplastics only exposure, rotenone only exposure, and the combination of rotenone and nanoplastics. We analyzed for mitochondrial content and mitochondrial coupling efficiencies. Let's look at the mitochondrial content first. Mitochondrial content was analyzed in whole larvae and separately in head, heart, and yolk sac. Each graph has 2 faces. Left-hand side is for nanoplastics only exposure and control is indicated in black color. Right-hand side is for combined exposures, rotenone only exposure indicated in blue color. Star signs indicated differences from the control, and hashtag is for significant differences between single and co-exposed counterparts. We see an increase in mitochondrial content with nanoplastic exposures. As expected, rotenone-only exposure has the highest mitochondrial content. Interestingly, at combined exposures, mitochondrial content was decreased, suggesting possible reduction of rotenone toxicity in the presence of nanoplastics. Moving on, we look at these contaminant effects on respiration, such as total basals and maximum respiration and the spare capacity. Again, each graph has 2 faces. Red color is allocated for nanoplastic only exposures, and the green color is for combined exposures. We didn't see any change in total basals with the nanoplastic exposure, and there's a significant decrease in total basals in the rotenone-only exposure group. Interestingly, we see an increase in total basal at the combined exposure, especially at the highest dose of nanoplastic and the rotenone, which is suggesting the reduction of rotenone toxicity in the presence of nanoplastic. We see the similar trend for the maximum respiration and the spare capacity. Concluding this work, we can say nanoplastics can accumulate in animal tissues. They target mitochondria and interfere with their function. They soak to lipophilic contaminants such as rotenone and modify mitochondrial toxicity of the rotenone. Does that mean nanoplastic can clean the animal? We have to do more research on that. Thank you.